So based on the previous video that I have made, once the L-fucose is attached, the structure is officially recognized as the H antigen. So why L-fucose matters? L-fucose is known as the immunodominant sugar for the H antigen. This means it is the specific molecule responsible for H specificity. Without this sugar, the structure cannot function as a foundation for the ABO system. So the H antigen is the mandatory acceptor molecule for the A and B genes. In group A individuals, the A gene enzyme adds another sugar, which is the N-acetylgalactosamine to this H foundation. In group B individuals, the B gene enzyme adds the galactose to this H foundation. In group O individuals, the H gene remains unmodified because the O gene is non-functional, leaving the red cells rich in H antigens. So again, the only antigen in the H blood group system, the O gene inherit at least, the blood group O inherit at least one H gene, or genotype big H, or the big H small h. So we have here the Bombay phenotype. If an individual inherits two rare small h alleles, therefore the genotype is small h, they cannot produce the necessary L-fucosyl transferase enzyme. So as a result, L-fucose is never added to the precursor chain. Because they lack this essential H foundation, they cannot express A or B antigens even if they have the A or B genes. This rare condition is again known as the Bombay phenotype. Now let's talk about it further. The Bombay phenotype is designated as big O small h. It is a rare genetic condition first discovered in 1952 in Bombay, India. So it is inherited as an autosomal recessive trait occurring when a person inherits two silent small h alleles. So the genotype of the person is small h instead of the common big H gene. This genotype is significant because it fails to produce the enzyme alpha-2 L-fucosyl transferase. This enzyme is required to transfer a specific sugar, L-fucose, onto a precursor chain to create the H antigen. Because the H antigen is the mandatory building block or the acceptor molecule for the ABO system, its absence means the individual cannot express A or B antigens on their red cells, even if they have inherited the A or B genes from their parents. In the laboratory, the Bombay phenotype presents the following characteristics. So they appear to be group O in forward and reverse grouping because they lack A and B antigens. And then, unlike normal group O red cells, which are rich in big H antigen, the Bombay red cells will not react with the anti-H lectin, which is Ulex europaeus. Their serum contains anti-A, anti-B, anti-AB, and a potential anti-big H antibody. So these individuals are non-secretors, meaning they do not have A, B, or H substances in their saliva or other body fluids. So the clinical implications for these individuals are severe because their serum contains a potent anti-H antibody that reacts strongly at body temperature. They are incompatible with group O blood. So transfusing group O blood into a Bombay recipient would cause immediate and potentially fatal intravascular hemolysis. Therefore, a person with a Bombay phenotype can only receive blood from another Bombay individual. Now let's proceed to the formation of the A antigen. So it includes a multi-step biochemical process that occurs on the red blood cell membrane governed by genetics and specific enzymatic activity. So the instructions for creating the A antigen are located again on chromosome 9 at the ABO locus. An individual inherits one ABO gene from each parent, and if at least one of those is an A gene, the body will produce the specific tools needed to build the A antigen. The A antigen cannot be formed from scratch, so it requires a pre-existing foundation called the H antigen. First, the H gene on chromosome 19 produces an enzyme that attaches a sugar called L-fucose to a precursor chain on the RBC. This structure is now the H antigen, which acts as the acceptor molecule for the A-specific sugar. The A gene does not produce the antigen directly. Instead, it codes for a specific glycosyltransferase enzyme known as alpha-3N-acetylgalactosaminil transferase. This enzyme's sole job is to recognize the H antigen foundation and build onto it. The A enzyme facilitates a chemical reaction that captures a specific simple sugar and attaches it to the H antigen. The specific molecule is the N-acetyl D-galactosamine or the GalNAc. So the enzyme transfers this sugar to the terminal galactose of the H-saturated precursor chain. Once the sugar is attached, the entire structure is transformed and is now recognized as the A antigen. Because this sugar is responsible for the unique identity of the blood type, it is called the immunodominant sugar. So the number of A antigens formed on a cell depends on the version of the A gene inherited. So the A1 gene is very potent, creating between 810,000 and 1,170,000 antigen sites on a single adult RBC. The A2 gene produces a less efficient enzyme, resulting in only about 240,000 to 290,000 sites. In individuals who inherit both A and B genes, the B enzyme is often more efficient at competing for the H foundation, meaning there are typically fewer A antigens than B antigens on the red cell surface. So, again, everyone, the genotype big A, or homozygous A, or heterozygous big A and then O. Those genes codes for the production of the enzyme alpha-3N-acetylgalactosaminil transferase, which transfers an acetyl D-galactosamine sugar to the H substance. And then that sugar is responsible for the A antigen expression, and the immunodominant sugar that is linked to the type 2 precursor substance now contains the H substance through the action of the H gene. Now let's proceed to the formation of B antigen. So it's a specific biochemical process that occurs on the red blood cells surface. So turning a basic precursor into a unique marker of blood group identity. Again, this process is controlled by genetics and the action of specialized enzymes. So the blueprint for the B antigen is the B gene, which is located on chromosome 9 again. So this gene is codominant, meaning if it is inherited from even one parent, the B gene will be expressed on the red cells. The B gene does not create the antigen directly. Instead, it codes for the production of a specific protein called an enzyme, alpha-3D galactosyltransferase. Think of this enzyme as a biological builder, whose only job is to attach to a specific sugar to a foundation. Before the B antigen can be built, the cell must have a foundation known as the H antigen. The H antigen is made from a type 2 precursor chain or the paragloboside already found on the red cell membrane. Without this H antigen foundation, the B enzyme has nowhere to attach its sugar, which is what happens in the rare Bombay phenotype. Once the builder or the enzyme and the foundation, which is the H antigen, are present, the final step occurs. So the alpha 3D galactosyl transferase enzyme facilitates the transfer of a simple sugar called D-galactose to the H antigen structure. So D-galactose is known as the immunodominant sugar for B specificity. This specific sugar molecule is what an antibody recognizes as the B blood type. Once the D-galactose is attached to the H foundation, the structure is officially the B antigen. So an adult with type B blood typically has between 600,000 and 830,000 B antigen sites on each red cell. Because the B enzyme uses up the H foundation to create B antigens, type B individuals have fewer leftover of H antigens than the type O individuals. So if you are wondering about the group AB individuals, so in people who inherit both A and B genes, the B enzyme is actually more efficient than the A enzyme at competing for the H foundation, often resulting in slightly more B antigens than A antigens on their red cell surfaces. So again, if an individual has the genotype homozygous B or heterozygous BO, so those genes codes for the production of alpha 3D galactosyl transferase, which attaches the immunodominant sugar D-galactose to the H antigen, which is responsible for the blood type B specificity. So this table is taken from our reference book. It only shows the different glycosyl transferases and immunodominant sugars responsible for the expression of H, A, and B antigens. So please don't forget to memorize this table. Now, let's proceed to the biochemical structures of your H, A, and B antigens. So the biochemical structures of A, B, and H antigens are built upon a common foundation known as the precursor substance, specifically a glycan or a paragloboside. So this structure consists of an oligosaccharide chain, which is a short string of 4 sugar molecules attached to either a protein or a lipid carrier molecule on the red cell membrane. On the red blood cells, these antigens are constructed on type 2 chains, which are defined by a specific beta 1,4 chemical linkage between the terminal sugars D-galactose and N-acetylglucosamine. So the H antigen is the mandatory precursor or the acceptor molecule required for the formation of A and B antigens. Its structure is determined by the H gene, which is located on chromosome 19, which produces a transferase enzyme that attaches a simple sugar called the L-fucose to the terminal galactose of the type 2 precursor chain. So the immunodominant sugar of the H antigen is L-fucose. It is the molecule responsible for H specificity. And take note, because the O gene is non-functional, it does not modify the H substance. Consequently, group O cells have the highest concentration of unmodified H antigens. Let's talk about the A antigen. It is formed when an individual inherits the A gene on chromosome 9, which codes for the production of the enzyme alpha-3-N-acetylgalactosaminil transferase. This enzyme recognizes the H antigen foundation and builds upon it by adding a specific sugar. So the enzyme transfers N-acetyl D-galactosamine, which is the immunodominant sugar, to the H structure. So an adult A1 RBC possesses approximately 810,000 to 1,170,000 antigen sites. So lastly, the B antigen is created when the B gene, also on chromosome 9, produces the enzyme alpha-3D galactosyl transferase. Like the A enzyme, it uses the H antigen as its starting point. So this enzyme attaches the immunodominant sugar D-galactose to the terminal galactose of the H-saturated precursor chain. So in individuals who inherit both A and B genes, or group AB, The B enzyme competes more efficiently for the H substance foundation, typically resulting in a slightly higher number of B antigens than the A antigens on the red cell surface. So again, just to give you another visualization of the biochemical structures of the A, B, and H antigen. So I have here some more so that you can really familiarize this. Now let's proceed to the molecular basis of ABO blood types. So according to Yamamoto et al., no, there are few mutations exist in the glycosyltransferase gene at the ABO locus. So the enzymes differ in amino acid composition. As you can see on the diagram presented on your screen. So the molecular basis of ABO blood types is centered on the genetic instructions located on chromosome 9. So these genes determine an individual's blood type by controlling the production of specific enzymes that build antigens on the red cell surface. So again, the A and B alleles do not create antigens directly. They code for proteins called glycosyltransferases. And then these enzymes act as biological builders that attach specific immunodominant sugars to a foundation molecule known as the H antigen. So the A gene, this allele codes for alpha-3N acetyl galactosamine transferase, which attaches the immunodominant sugar and acetyl D-galactosamine to the H antigen. The B gene, this allele codes for an alpha-3D galactosyl transferase enzyme, which attaches the sugar D-galactose to the H antigen. And then the O gene, this allele is an amorph or a silent gene, meaning it does not produce a functional enzyme. Because the H antigen remains unmodified, type O individuals have the highest concentration of H substance on their red cells. Now, ABO blood group antigens exists in two forms, as insoluble glycolipids on the surface of red blood cells, and as soluble glycoproteins found in body fluids and secretions. While most people consider blood type to be a feature of the blood, these antigens are widely distributed on platelets, lymphocytes, epithelial cells, and organs like the kidneys. So let's talk about the role of the secretor gene, which is written in the symbol capital S, small e. So the role of your secretor gene, the ability to produce soluble ABH antigens, is controlled by the secretor gene, also known as your FUT2, which is located on chromosome 19. This gene is inherited independently of the ABO and H genes. Individuals who inherit at least one functional capital S, small e gene, let's say for instance the genotype is big S, small e, big S, small e, or big S, small e, small s, small e, are classified as secretors. So these individuals express soluble H antigens in their secretions, which can then be converted into soluble A or B antigens depending on their ABO genotype. Individuals who inherit two silent alleles, therefore the genotype is small se, are non-secretors. They do not produce the necessary enzyme to form soluble H substance, and therefore their secretions contain no soluble A, B, or H antigens. So the primary biochemical difference between RBC-bound antigens and soluble antigens is the precursor chain they are built upon. Again, type 2 chains are found in your RBCs. So antigens on the red cell membrane are primarily built on a type 2 oligosaccharide chain defined by a beta-1,4 linkage. While your soluble antigens in the body fluids like saliva, tears, and milk are primarily built on type 1 oligosaccharide chains defined by a beta 1,3 linkage. So in a secretor, the SE gene codes for the enzyme alpha 2L fucosyl transferase, so which adds the sugar L-fucose to the type 1 precursor chain. This creates a soluble H substance. Once this H base is present, the individual's ABO genes can then add their specific immunodominant sugars and acetyl D-galactosamine for A specificity or D-galactose for B specificity to create soluble A or B antigens. Soluble A, B, H antigens can be detected in almost all body fluids, including saliva, urine, tears, amniotic fluid, and breast milk. But they are notably absent from the cerebrospinal fluid. The specific substances found in the saliva of secretors depend entirely on their ABO blood group. Group O secretors, they only contain the H substance. Group A secretors, they have A and H substances. For Group B secretors, they have the B and H substances. For Group AB secretors, they have the A, B, and H substances. Understanding secretor status is valuable in their laboratory for resolving ABO discrepancies. In cases where a patient's RBC antigens are weakly developed or difficult to detect, a heme agglutination inhibition test on their saliva can confirm their true ABO group by identifying the soluble antigens they secrete. Furthermore, the secretor gene or the SE gene is closely linked to the big H and Lutheran genes, and its absence is a defining feature of the rare Bombay phenotype, or the small h, as these individuals lack the big H foundation on both their cells and in their secretions. So again, the secretor genes could be the homozygous big SE, or the heterozygous big SE, small SE. And then you are a non-secretor if you have the homozygous small SE, small SE. Blood group antigens are found in various locations throughout the body, ranging from structural components of the red cell membrane to soluble forms in the body fluids. So based on the our references, here's quite an explanation of their locations. So your antigens are integral parts of the cell membrane, so meaning they are intrinsic or integral portions of the cell membrane. So these integral antigens are typically large molecular weight proteins such as the RH system, glycoproteins or proteins coupled with carbohydrates like the CAL system, or glycolipids, carbohydrates coupled with lipids like the ABO system. So they often traverse the lipid bilayer multiple times. So for example, the RH system proteins are multipass proteins that loop through the membrane 12 times. So because these antigens are integral parts of the membrane structure, they often protrude from the surface in three-dimensional configurations, making them accessible to antibodies. So RBCs are the primary location for blood group antigens, where they form part of the lipid bilayer or extend from the surface. So some systems such as the RH and CAL are considered confined almost exclusively to the red cell membrane, while antigens like ABO are detectable as early as 5 to 6 weeks in utero. They are not fully developed at birth. Newborns possess fewer antigen copies and partially developed structures compared to adults. So endothelial cells, which are the cells lining the blood vessels, also express certain blood group antigens. Platelets express several types of antigens that are critical for transfusion and transplantation compatibility. Lymphocytes, like other nucleated cells, express antigens primarily related to the HLA system. And then epithelial cells across various organs express a variety of blood group markers. Now let's try to apply your knowledge from the previous discussion that we have on uh, if the antigens that you have inherited can be expressed on either the RBC or saliva. So we have here the first example, the genes inherited on this first person. We have the AB, then the big H, then the big SE. Second example, we have the big AB, the big H, then small se. And then we have the big O, the homozygous O, then the big H, then big SE, small se. And then lastly, the big O, and then big H, and then the small se. So now I will reveal the answers to you. So here it is. You can pause the video if you like. So the expression of blood group antigens on the red blood cells and in secretions like saliva depends on the interaction of the three independently inherited sets of genes: the ABO, the big H, small h, and the secretor gene. So the H gene or the FUT1 produces the foundation H antigen on RBCs, while the SE gene or the so-called FUT2 produces the foundation for soluble antigens in saliva. The A and B genes then code for specific transferase enzymes that add immunodominant sugars to these H foundations. So based on the genetic combinations provided on our example, the expressions are as follows. For the first example, AB, big H, and then the homozygous big SE. So the presence of the H gene, big H, ensures the formation of the H antigen on type 2 precursor chains on the RBC membrane. So the A and B genes then produce transferases that convert the H foundation into A and B antigens. Because the B enzyme is often more efficient, some unconverted H remains. So since the individual is a homozygous secretor, the SE gene produces the enzyme needed to form the soluble H substances in the secretions. So the A and B genes then modify the soluble H into a soluble A and B substances. For the second example, the individual is a heterozygous for the H gene, so meaning it has a big H and small h, which is sufficient to produce the H antigen foundation on the RBCs. This also allows the A and B genes to express the A and B antigens normally on the red cell surface. The individual is a non-secretor. Why? Because the genotype se or the se allele is an amorph, meaning it fails to produce the enzyme necessary to create the H substance foundation in secretions. Without this H foundation, the A and B genes have no acceptor molecule to work with, resulting in no detectable ABH substances in the saliva. The third example, with functional H gene, big H, the H foundation is built on the RBCs. However, the O gene is an amorph or non-functional and does not produce an active transferase to add further sugars. Consequently, the H antigen remains unmodified and group O cells have the highest concentration of H antigen. So, as for the expression in the saliva, the individual is a heterozygous secretor, which contains the genotype big SE, small se, meaning they can produce the soluble H foundation in their secretions. Because they lack A or B genes, the H substance is not converted into A or B substances, leaving only soluble H detectable in the saliva. For the last example, similar to the case above, the functional H genes creates the H antigen on the RBCs. Because only O genes are inherited, the H antigen remains unmodified and is the only antigen expressed on the red blood cells. So despite having functional H and O genes, the individual is a non-secretor, meaning it has the genotype se. They cannot form the necessary soluble H substance foundation in their body fluids. Therefore, no ABH substances are expressed in the saliva. Again, just a quick recap of the ABO antibodies. They are produced without any exposure to RBCs, meaning they are naturally occurring. They are predominantly IgM in nature. They activate the complement and react at room temperature or colder. And then they produce strong agglutination during testing, usually at 3 plus to 4 plus. Production is initiated at birth. Titers are generally low or too low for detection until individual is 3 to 6 months of age. So antibodies found in the cord blood serum are of maternal in origin. And if ever wrong ABO blood group is transfused to the patient, then it will cause intravascular hemolysis. So again, these are the possible combinations of the ABO blood groups of the offspring of different ABO matings. Now let's talk about forward grouping, also known as the front typing. It is defined using known sources of commercial anti-sera, specifically anti-A and anti-B, to detect the presence or absence of A and B antigens on a patient's red blood cells. So this is the most frequently performed test in the blood bank, and it's required for all donors and patients to ensure transfusion safety. So let's talk about the slide method. So while the tube test is the routine standard for manual testing, the slide method is another traditional technique utilized in certain settings. So commercial anti-A and anti-B reagents are typically labeled by the manufacturer for use in slide, tube, and as well as microplate testing according to their FDA licensure. So in this method, a drop of the patient's blood is mixed with a drop of the appropriate anti-sera on a glass slide or tile. So you place the anti-sera, add the patient's red cells, and then after that, you are going to read the results. Okay? So now let's talk about the reverse typing. It's also known as the reverse grouping or back typing or ABO serum testing. It is part of an ABO blood group test used to detect your ABO antibodies or your isoagglutinins in a patient's serum or plasma. It follows Landsteiner's rule, which states that individuals predictably possess antibodies in their serum directed against the ABO antigens absent from their own red blood cells. So while the sources, our references describe the tube method as a standard manual technique for this procedure, they also, our references also mention that ABO reagents are frequently labeled for slide use. However, they highlight that the tube method and newer automated methods like the gel and the solid phase are the primary ways to perform these tests in a clinical setting. So although our references primarily detail the tube method for reverse typing due to its higher sensitivity, our references also note that ABO reagents primarily used for forward grouping are often labeled for slide, tube, and microplate testing. So historically, slide methods were used in emergency or field settings. So in a slide method, you're going to put a drop of the patient's serum and then it's mixed with a drop of reagent. Could be A1 or B cells on the slide, and then the slide is rotated or tilted to allow for the interaction of antigens and antibodies. So agglutination is observed macroscopically, and then this you need to take note that tube testing is more reliable because centrifugation brings the antigens and antibodies into closer physical proximity, allowing them to overcome the zeta potential or the repulsive repulsive forces of the RBCs more effectively than on the slide. So observation for the slide method for forward typing. The mixture is observed for the presence of or absence of visible clamping or agglutination of the red cells. So if there is a positive reaction, it indicates the presence of agglutination or hemolysis. It indicates that the corresponding antigen is present on the patient's red cells. For the reaction strength, the routine ABO forward typing reagents are highly potent and usually demonstrate strong reactions, typically 3 plus to 4 plus in strength. So for example, if a patient's cell clump with anti-A but not anti-B, they possess the A antigen and are classified as group A. If no clumping occurs with either reagent, the cells lack both antigens and are classified as group O. So the forward grouping must always be confirmed by a reverse grouping, or detecting the antibodies in the patient's serum to ensure the results match Landsteiner's rule. So here is for the slide method of the reverse typing. So again, you need to follow the Landsteiner's law or the Landsteiner's rule. Now let's talk about the forward typing, the tube method. It's a traditional manual technique used to detect antigens through the following standardized steps. So the medical technologies first label the test tubes and prepare a 2 to 5% suspension of the patient's red cells in saline. So 1 drop of commercial anti-A, which contains a blue dye, is added to the first tube, and 1 drop of anti-B, which contains a yellow dye, is added to the second tube. So 1 drop of the prepared patient's RBC suspension is added to each reagent tube. The tubes are mixed and then centrifuged for approximately 20 seconds at a calibrated speed. So the technologist gently shakes and tilts the tube to dislodge the cell button, observing for agglutination or clumping or hemolysis. Now let's talk about the reverse typing tube method. It's the traditional manual technique for reverse grouping because ABO antibodies are primarily of the IgM class. So they react optimally in the immediate spin phase at room temperature, usually 20 degrees Celsius to 24 degrees Celsius. So the same procedure, the medical technologists will label 2 test tubes, such as the A1 or the B cells. Then you're going to add 2 drops of the patient's serum or plasma to each tube, and then 1 drop of known commercial reagent red cell, either a 2 to 5% suspension of A1 and B cells. And then the tubes are mixed and centrifuged, typically for approximately 20 seconds at a calibrated speed. And then the cell button is gently resuspended by tilting or rolling the tube. So you're going to observe for visible clamping of red cells. You can grade it as 0 to 4 plus. And then if there is hemolysis, you can observe pink or red discoloration of the supernatant, which is also interpreted as a positive reaction. So for a valid ABO type, the reverse grouping must inversely match the forward grouping. For example, a group A individual serum should agglutinate B cells but not A1 cells. So these are just different reactions of the tube method using the forward and the reverse typing. So in here, you can also see that there is an agglutination because your antigens from the red cells binded with the antibodies present. So which will now result into this. So in the tube method, antigen-antibody reactions are measured qualitatively by observing the degree of red cell clumping or agglutination, or the rupture of red cells, known as your hemolysis. So the standardized grading system used to record these observations follows a scale of 0 to 4 plus. So to determine the grade, the medical technologist should Gently shake and tilt the test tube until the red cell button is completely dislodged from the bottom. So the reactions can be defined as 4 plus. It's characterized by one solid agglutinate or one solid clump and a clear background. So 3 plus consists of several large agglutinates with a clear background. 2 plus shows many medium-sized agglutinates and a clear background. 1 plus reaction, it can be defined by medium and small-sized agglutinates in a turbid or a cloudy background containing many free and agglutinated red cells. We also have W plus, or known as the weak reaction. It displays tiny agglutinates that are barely visible to the naked eye in a turbid background. We also have the zero, or the negative. No agglutinated cells are visible. Then the red cells flow off the button in a smooth suspension during the grading process. So here it is. No, so aside from those that I have mentioned, 0, 1 plus, 2 plus, 3 plus, 4 plus, and then W plus. So we have also other positive indicators. We have hemolysis, in which a pink or red discoloration of the supernatant or the liquid above your red cells after centrifugation is interpreted as a positive reaction. So this occurs when the complement system is activated, causing the red cell membranes to rupture. We also have small m, small f, or the so-called mixed field reaction. This describes a specific pattern wherein a population of agglutinated cells is seen alongside a mass of unagglutinated cells. This is common in patients who have recently received a non-ABO identical transfusion, such as group O cells given to a group B patient. Now let's talk about the gel technology or the gel method. It's also known as a column agglutination technology or the CAT. It's a method of blood typing that uses a plastic card containing specially designed microtubes to detect antigen-antibody reactions. So it is developed to standardize traditional testing. This method now eliminates the need for manual tube shaking to resuspend red cell buttons, providing more objective and reproducible results. So for the principle, each microtube in a gel card contains dextran acrylamide gel particles that act as a molecular sieve. During controlled centrifugation, large agglutinates are trapped at the top of the gel, while smaller agglutinates travel further down. Unagglutinated or negative red blood cells pass through the gel entirely and form a solid pellet at the very bottom of the microtube. So forward typing in gel is used to detect the ABO and RH antigens on a patient's red blood cells. So the manufacturer preloads specific microtubes with gel containing monoclonal anti-sera such as anti-A, anti-B, and anti-D. As presented in your screen, so it has labels. So a 4% suspension of the patient's red cells is prepared in a specific diluent, and then 10 microliters of this suspension is pipetted into the reaction chamber of the appropriate microtubes. So the card is centrifuged for 10 minutes at a calibrated speed. So agglutinated cells trapped at the top or within the gel column indicates a positive reaction for that antigen. For the reverse typing in gel, it detects ABO antibodies in a patient's serum or plasma. So the test uses microtubes containing neutral buffered gel without added antibodies. So known reagent RBCs such as your A1 cells and the B cells are used as the antigen source. You are going to get 50 microliters of 0.8% reagent red cells. You're going to add it to the microtubes and then followed by a 25 UL of the patient's serum or plasma. So the card is typically centrifuged for 10 minutes. So clumping in the gel column indicates the presence of the corresponding ABO antibodies. But again, the volume of the reagent and the sample varies from one manufacturer to another. So how are we going to grade the results of our gel method? So the position of the red cells in the microtube determines the grade of the reaction. So 4 plus, a solid band of red cells at the top of the gel. 3 plus, agglutinates located in the upper half of the gel column. 2 plus, agglutinates dispersed throughout the entire length of the column. And 1 plus, agglutinates predominantly in the lower half of the column. 0 or negative, a well-defined cell button at the bottom of the microtube with a clear gel above it. We also have MF, or the mixed field, a layer of cells at the top of the gel with a distinct pellet of cells at the bottom. So this picture shows the formation of H antigen. It is genetically controlled by the H gene, or the FUT1, located on chromosome 19. So this gene codes for the production of the glycosyltransferase enzyme, alpha-2 L-fucosyltransferase, or also called as your L-fucosyltransferase. This enzyme catalyzes the transfer of the immunodominant sugar L-fucose to the terminal galactose of a type 2 precursor substance or oligosaccharide chain. This step is essential because the H antigen serves as the mandatory acceptor molecule, or the foundation upon which the A and B antigens are built. So the formation of your A antigen requires a mandatory foundation known as the H antigen. So the A gene, located on chromosome 9, codes for the production of a specific glycosyltransferase enzyme called alpha-3N acetylgalactosaminil transferase. This enzyme catalyzes the transfer of the immunodominant sugar and acetyl D-galactosamine, or the GAL and AC, to the terminal galactose of the H saturated precursor chain. Once the sugar is attached, the entire biochemical structure is transformed and officially recognized as the A antigen. So now we have here the formation of the B antigen. Its biochemical process is controlled by the B gene located on the chromosome 9. So this gene codes for the production of a specific glycosyltransferase enzyme known as the alpha-3D galactosyltransferase. So this enzyme catalyzes the attachment of the immunodominant sugar, D-galactose, to the terminal galactose of the pre-existing H antigen foundation on the red cell membrane. So once this specific sugar is added, the structure is transformed and recognized as a functional B antigen. This image shows a carbohydrate or a glycan chain that's characteristic of the ABO blood group antigen precursor, specifically the H antigen backbone. So it's a branch oligosaccharide attached to a carrier labeled as R, which represents a glycogen, or a glycoprotein or a glycolipid on the red blood cell membrane. So the colored hexagons are monosaccharides, and then the sugar components from the reducing end outward. So you have JL, or the galactose, its multiple galactose units form the backbone. The GLC and AC, or the N-acetylglucosamine, it's a key structural sugar in ABO antigen chains. You have your FUC, or the fucose, attached as a side branch. So the fucose linked to a galactose is the defining feature of your H antigen. And then the H antigen is the precursor substrate for A antigen. When N-acetylgalactosamine is added, then the B antigen when galactose is added. So if no additional sugar is added, then the structure remains blood group O, or H antigen only. As for the blood type AB individuals, their red cell contains the A and B antigens. Next, I have here the different combinations of your ABH formations, and upon which they have added a specific immunodominant sugar. And then here I have a summary of the tables showing the different reactions of your blood group A individuals, the B individuals, and then the AB, and as well as the blood group O. So kindly familiarize this, everyone. And lastly, that would be the end of my discussion. So hopefully you have learned something from this video. So stay safe, and once again, have a great day.